Welcome back to the deep dive. Today we're tackling something huge, the whole regulatory landscape of the eukaryotic cell. And we're starting with, well, a truly remarkable survivor. You might even call it a microscopic terrorist.
We're diving into Trypanosoma brucei. That's the parasite behind sleeping sickness and Nagana. And the reason it's so incredibly successful is basically a master of molecular evasion.
It really has to be, right? Because the moment it gets into a host, the immune system is immediately on high alert. It's ready to make antibodies against, well, whatever foreign stuff it finds. And in this case, the parasite is covered, completely coated, in just one protein type, the variant surface glycoprotein, or VSG.
Right, so it's an obvious target. The immune system launches this powerful attack, clears out maybe 99% of the parasites with that specific VSG coat. But here's the kicker. Before the immune response wipes everyone out, a few parasites in the population somehow manage to switch. They swap out that entire protein coat for a completely different VSG type, one the immune system hasn't seen before.
That's just incredible. It's like biological improvisation on the fly. It means they must have this huge library of molecular disguises ready to go. And crucially, a system to make sure only one disguise is active at a time, while hundreds of others are kept silent.
Exactly. And that needs incredibly precise, time-sensitive regulation, temporal regulation. It's actually the perfect starting point for our deep dive today, because complex life, from parasites right up to us, needs genes regulated not just in time, but also in space. How does your liver cell know it's a liver cell and not, say, a neuron?
Okay, yeah. That's the mission then. To walk you through the main control points, we're talking 4 key levels, right? From the DNA sequence itself all the way up to these big structural changes.
That's the plan.
All right, let's unpack this. Eukaryotic gene regulation is just inherently more complicated than in bacteria. And the main reason, our sources emphasize, is compartmentalization, the cell's internal structure.
Right, that physical separation changes everything. Transcription, making the RNA message, and then processing it, capping, adding the poly-A tail, splicing out introns. That all happens inside the nucleus.
Right.
But translation, the actual protein building, that happens out in the cytoplasm.
So the cell's got multiple hurdles. It has to regulate the initial transcription, and then it has to control the RNA's journey out, how long it survives, and whether it gets translated. That's a lot of checkpoints.
It is. Let's start at the beginning, though. Inside the nucleus. Transcriptional control. How does the cell actually respond to signals from the environment and, flip those gene switches?
Well, a really classic example, one that shows this beautifully, is the heat shock response. You see this in fruit flies, Drosophila. When things get too hot, say above 33 Celsius, the cell needs to protect itself fast.
Yeah, it kicks off this rapid program to transcribe protective genes. The main one is heat shock protein 70, or HSP70. Basically, the cell translates that environmental stress, the heat, into a chemical signal, and that signal activates a key regulatory protein.
And how does that activation work? What's the mechanism?
Well, the heat stress leads to a chemical modification, usually phosphorylation, of the heat shock transcription factor, or HSTF. Once it's modified, this HSTF can then bind to very specific short DNA sequences, usually upstream of those protective genes.
And those are the heat shock response elements, HSEs.
Exactly, the HSEs. That binding is the crucial signal. It basically tells RNA polymerase, okay, go time, transcribe HSP70.
Okay, so environmental stress gets translated into specific gene activation. We see something similar with hormones, right? But they seem to use 2 pretty different strategies to deliver their messages. Let's start with the ones like estrogen, the lipid-soluble ones.
Ah, right, the steroid hormones. Because they're lipids, they're fatty, they don't need a special doorway, they can just, cruise right through the cell membrane, into the cytoplasm, or sometimes even straight into the nucleus.
And once they're inside, how do they actually issue a command to the genes?
They find and bind to a specific receptor protein. And this receptor is often just kind of hanging out, inactive, until the hormone docks with it. The hormone receptor complex becomes the active transcription factor. It then goes to the DNA, finds specific sequences called hormone response elements, or HREs, binds there, and directly stimulates transcription. It's a very direct, elegant system.
Okay, that's the direct route.
Right.
Now, what about the other kind, the big ones like insulin, peptide hormones. They're
Whoa.
too large to just slip through the membrane.
They definitely are. So they use signal transduction. Insulin binds to a receptor that's embedded in the cell membrane on the outside surface.
Because the signal starts outside.
Right. That binding event triggers a kind of internal relay race, a cascade of chemical signals involving kinases, second messengers, all sorts of things, transmitting the signal across the cytoplasm. And eventually, that message activates a transcription factor waiting inside the nucleus. The receptor itself stays outside, but the message gets through.
That's a key distinction. Okay, so regardless of how the signal arrives, heat, steroid, peptide, hormone, we keep coming back to transcription factors binding DNA, which brings us to the sort of molecular architecture around the gene itself. We know the promoter is where RNA polymerase lands, but what about those other regulatory sequences, the really flexible ones?
You mean enhancers. Promoters are usually right next to the gene start site, very position and orientation dependent. Enhancers, though, they're different beasts. They're DNA sequences recognized by specific transcription factors, and they have these 3 classic properties.
Okay, what are they?
1, they can work over huge distances. We're talking tens of thousands of base pairs away from the gene they regulate. 2, their orientation doesn't matter, flipping backwards, they still work. And 3, their position is flexible, they can be upstream, downstream, even hiding inside an intron.
Hang on. If a transcription factor binds way over there, thousands of bases away, how does it physically talk to the RNA polymerase sitting at the promoter? That seems far.
Yeah, it's too far for just shouting. This is where the physical nature of DNA becomes critical. The DNA itself has to bend. And the thing that makes this possible, this large-scale interaction, is called the mediator complex.
The mediator complex. I like that name. Sounds like a molecular go-between.
That's exactly what it is. It's this huge assembly of proteins, like a massive molecular bridge. It physically connects the transcription factors bound way out at the enhancer to the machinery, the basal transcription factors, and RNA polymerase sitting at the promoter. It literally loops the intervening DNA out of the way to make contact.
Wow, that's some serious molecular scaffolding. And speaking of molecular shapes, let's look at the transcription factors themselves. They aren't just formless blobs, are they? They have specific structures, motifs, that let them actually grab onto DNA.
Absolutely. They have characteristic shapes. One very common one is the zinc finger. It's a little loop of protein, a peptide loop that gets stabilized when cysteine and histidine residues coordinate a zinc ion. This stabilized finger-like structure then juts out and can probe the DNA grooves for specific sequences.
And then there's the helix-turn-helix motif. 3 short helices, right? That one's often part of the homeodomain, which is super important in development. The sources mention those really weird cases, homeotic transformations, like a fly growing legs where its antenna should...
Yeah, that's a dramatic failure of developmental control, precisely because these transcription factors aren't working correctly. And we also have motifs designed for dimerization for 2 protein chains to pair up.
Like the leucine zipper.
Exactly, the leucine zipper. You have these stretches where leucine amino acids appear every 7th position. When 2 of these protein regions come together, the leucines interdigitate, like a zipper, holding the 2 polypeptides together as a dimer. And adjacent regions, often positively charged, are then perfectly positioned to bind the negatively charged DNA backbone.
And there's a similar one, too, the helix-loop-helix. 2 helices separated by a loop, also for dimerization.
That's right. And the really powerful idea here, especially with dimerization, is the potential for mixing and matching. You can form homodimers, 2 identical partners, or heterodimers, 2 different partners.
So combining different factors.
Precisely. Heterodimer formation gives you this incredible combinatorial complexity. Instead of just simple on-off switches, you get a whole sophisticated switchboard. Mixing different partners allows for much more subtle, fine-tuned, tissue-specific patterns of gene expression. It's really elegant.
That makes sense. Much more nuanced than just yes or no transcription. But, okay, regulation doesn't just stop once the RNA is made, right? There's a whole other layer. Let's talk about post-transcriptional control, starting with splicing.
Alternate splicing. Yeah, this is a fantastic example of how eukaryotes get more bang for their buck, genetically speaking. Most eukaryotic genes have introns, these non-coding bits interspersed with exons, the coding bits. The splicing machinery can choose to include or exclude certain exons when it stitches the mature mRNA together.
And the power that gives the cell is just huge. The example from the source, the ret troponin T gene, it has 18 exons, but through alternate splicing, it can generate something like 64 different mRNAs, all from one gene.
Exactly, leading to 64 distinct though related, protein versions. It means one gene can do the work of dozens. It's incredible economy.
Essential economy, I bet. Okay, so after splicing, the mRNA gets shipped out to the cytoplasm. What's the next control point?
Its lifespan, mRNA stability. An mRNA molecule that sticks around longer can be translated many times, producing a lot more protein than one that gets degraded quickly.
And what determines how long it lasts?
Several things. The length of the poly-A tail at the end is generally a factor. Longer tails often mean longer life. But there are also specific sequences within the mRNA, particularly in the 3' untranslated region, the 3' UTR, that act like timers or signals.
The countdown clocks.
Sort of. For instance, repeated AUUA sequences in the 3' UTR are known signals for rapid degradation. They basically flag the mRNA for destruction.
Okay, now let's get to something relatively recent but really exciting in regulation. RNA interference, RNAi, using tiny RNAs to silence genes.
Yes, RNAi. It's a fundamental mechanism found pretty much everywhere in eukaryotes. Its main job seems to be defense, fighting off viruses and jumping genes, transposons, but it's also a sophisticated layer of gene regulation itself. It all kicks off with an enzyme called dicer.
Dicer, like it dices things up. The molecular scissors.
That's a great way to think of it. It finds larger double-stranded RNA molecules and chops them up into these tiny pieces, typically 21 to 28 nucleotides long. These are the small interfering RNAs, siRNAs, or microRNAs, miRNAs.
Okay, tiny RNA fragments. What happens then?
These little RNAs get loaded into a protein complex called RISC. That's the RNA-Induced Silencing Complex. Inside RISC, the small RNA becomes single-stranded. Now it's basically a guide, ready to find a matching mRNA sequence in the cytoplasm.
And what happens when it finds its target depends on how well it matches, right?
Exactly. If the small RNA, usually in siRNA, pairs perfectly with the target mRNA sequence, a protein within RISC called Argonaute acts like a slicer. It cleaves the mRNA, cuts it in half.
And cut mRNA is useless.
Useless and quickly degraded. That's the knockout effect, often used for defense. Very effective. But if the pairing is imperfect, which is often the case with miRNAs, RISC still binds, but Argonaute doesn't cleave. Instead, the bound RISC complex just sits there and physically blocks translation.
So it doesn't destroy the message, just stops it from being read.
Right, it represses protein production more subtly. And this ability to specifically knock down gene expression is why RNAi has become such an incredibly powerful tool for researchers.
Okay, that's both transcriptional. Now let's go even deeper, to the most fundamental level of control. The structure of the DNA packaging itself, chromatin organization.
Yes, this is regulation by essentially accessibility, how tightly packed the DNA is. We generally talk about two main states. There's euchromatin that's loosely packed, accessible, generally where active genes are found.
And the opposite.
Heterochromatin, densely packed, often full of repetitive DNA sequences, and generally transcriptionally silent. Genes stuck in heterochromatin are typically switched off.
And the power of this packaging is shown by that phenomenon, position effect variegation, PEV.
Exactly. A classic example. If you have a gene that's normally active, like the white gene for eye color in Drosophila, and through some rearrangement it gets moved next to a block of heterochromatin, its expression becomes unstable.
Unstable how?
In some cells, the heterochromatin structure spreads and silences the gene. In others, it remains active. The result is a patchy or mottled eye color. It's variegation caused by the gene's position.
And that's key because it's epigenetic, right? The DNA sequence of the white gene hasn't changed at all, but its activity state on or off is being inherited by daughter cells based on the local chromatin structure.
Precisely. It's a heritable change, but not in the DNA sequence itself. And we can actually measure this openness. Active DNA in euchromatin is physically more exposed, making it more sensitive to digestion by an enzyme called DNase I. We look for DNase hypersensitive sites.
And these hypersensitive sites often mark important regulatory regions.
Very often. They pinpoint where key regulatory complexes bind. A great example is the locus control region, or LCR.
The LCR, that sounds important.
It is. It's often found far upstream of a whole cluster of related genes, like the human beta-globin genes, which make parts of our hemoglobin. The LCR acts like a master switch, ensuring the whole region is open and ready for expression at the right time. It's crucial for that developmental switch, making embryonic hemoglobin first, then fetal, then adult hemoglobin as we grow.
Wow. So this packaging isn't set in stone. It can be changed.
Oh, absolutely. It's highly dynamic. It's actively managed by processes we call chromatin remodeling.
Okay, so who are the remodelers? What enzymes are involved?
Well, a major way to control chromatin is by modifying the histone proteins that the DNA wraps around. Enzymes called histone acetyltransferases, or HATs, add acetyl groups to histones. This usually neutralizes some positive charge on the histones, loosening their grip on the negatively charged DNA.
Making it more accessible.
Generally, yes. Acetylation correlates strongly with active transcription.
So what is the opposite? Tightens things up.
That would be histone deacetylases, HDACs, which remove acetyl groups, and also histone methyltransferases, HMTs, which add methyl groups to histones. These modifications often lead to more condensed, silent chromatin. Plus, there are large protein machines, like the SNF complex, that use ATK energy to physically slide or even evict nucleosomes to expose underlying DNA sequences.
Okay, histone modifications. Is there anything else acting directly on the DNA itself for this structural control?
Yes. DNA methylation. This is the addition of a methyl group directly onto a cytosine base, usually when it's next to a guanine, the CpG dinucleotide.
And methylation is generally bad news for gene expression.
Generally repressive, yes. High levels of DNA methylation are strongly associated with silenced genes. We see extensive methylation on the inactive X chromosome in female mammals, for instance.
Ah, methylation. And that's absolutely central to probably the weirdest epigenetic thing, imprinting.
Oh, imprinting is fascinating. It means that for certain genes, whether the gene is expressed or not depends entirely on which parent you inherited it from.
So the same gene sequence, but it behaves differently if it came from mom versus dad.
Exactly. The classic example is the Igf2 gene in mice, coding for insulin-like growth factor 2. It's only expressed from the copy inherited from the father. The copy from the mother is silenced.
How does the cell even know?
The imprint is laid down during egg and sperm formation. It involves sex-specific patterns of DNA methylation. These patterns are established in the germline and then faithfully copied in all the somatic cells of the offspring, marking that chromosome's parental origin.
It's incredible this cell has that kind of memory. And this idea of controlling whole chromosomes brings us to a really fundamental problem for organisms with sex chromosomes, dosage compensation.
Right. If you have, say, females with 2 X chromosomes, XX, and males with 1 X and 1 Y, XY, you've got a potential dosage problem. Females have twice as many copies of all the genes on the X chromosome. You need a way to equalize the output of those X-linked genes between the sexes.
So everyone gets roughly the same amount of X gene products. How do different organisms solve this?
Evolution's come up with at least 3 distinct, really cool solutions. In mammals, like us, the strategy is inactivation.
Random X inactivation in females.
Exactly. Early in development, each cell in a female embryo randomly chooses 1 X chromosome to shut down almost completely. This is mediated by a remarkable long non-coding RNA called XES, which literally coats the chosen X chromosome and triggers its silencing and condensation into a bar body.
Okay, so mammals silence 1 X in females. What about fruit flies, Drosophila?
They do the opposite. They use hyperactivation. The single X chromosome in males gets its gene expression turned up, essentially doubled, to match the output from the 2 X's in females.
How do they turn it up?
There's a specific complex of proteins, the MSL proteins, male-specific lethal, and some non-coding RNAs called RHO-X RNAs that specifically binds along the length of the male X chromosome and boosts its transcription.
Wow, okay. So inactivation in mammals, hyperactivation in flies. Is there a third way?
There is. In the nematode worm C. elegans, they use hypoactivation. In the hermaphrodites, which are XX, both X chromosomes have their overall transcription levels turned down, partially repressed, so their combined output matches the output from the single X in the XO males.
Incredible. So shutting 1 down, turning 1 up, or turning 2 down slightly, 3 totally different mechanisms, all achieving the same biological outcome, balanced X chromosome gene dosage.
It really underscores the point, doesn't it? Gene regulation isn't just one lock and one key. It's this incredibly complex, multi-layered system. Controls acting at the level of transcription initiation, RNA processing, stability translation, and overlaid on all of that, this deep structural control through chromatin and chromosome-wide mechanisms.
What an amazing journey through the eukaryotic cell's regulatory toolkit. We've seen how signals from the environment trigger transcription, the clever architecture of enhancers and transcription factors, how the cell gets more from its genes with alternate splicing, the surveillance and silencing by RNAi, and finally, these profound structural controls, chromatin states, methylation, and even whole chromosome adjustments.
And when you think about those epigenetic layers, the chromatin modifications, the DNA methylation, the imprinting, X inactivation, the key thing is that these states, these patterns of on and off, are often heritable through cell division. The DNA sequence is the fundamental script, sure, but this structural memory dictates how that script gets interpreted in different cells and tissues.
Which leaves us with a really fascinating question for you to ponder. We know this cellular memory is crucial for our own development and cell identity. But how persistent is it? Can these non-sequence-based epigenetic states potentially span generations? And what would that mean for how we think about inheritance, perhaps going beyond just the letters of the DNA code?
Yeah, definitely something to think about.
Food for thought indeed.
Yeah.
Thank you for taking this deep dive with us today.